Welcoming everyone. This is Murat. I'm the CEO and co-founder of Magnimind Academy. I'm also a Stanford scientist. I do a lot of work in AI and medicine, a lot of projects with Chirag, and especially in brain cancer in general. And we do work with several other diseases as well. And as a Magnimind Academy, we've been organizing these meetups. We have 7 groups, over 12,000 members. And we have YouTube channel, which has a lot of good, informative, enriching talks, lectures, mini boot camps. I recommend people to surf through it. You'll find a lot of resonating talks. And yeah, we have 2 starting programs that's about data science boot camps. You can just learn about it when our friends share it. And I will let Chirag to introduce himself and our today's speaker, James. If just one announcement that please write your questions in Q&A, and that in chat it is getting lost mostly. And yeah, the Q&A will be better. Now, Chirag, is your turn.
Okay, thanks, Murat. Well, welcome everyone. And as Murat mentioned, my name is Chirag Patel. I'm a clinical assistant professor in the departments of neurology and radiology at Stanford. My practice is a neuro-oncologist at the Cancer Center. And as Murat mentioned, we've trained together here as postdocs in the radiology department and now work together on various projects related to glioblastoma, which is a form of brain cancer. So we're really thankful to have James joining us today as our guest. And so just as a way of introduction, Dr. James Zou is an assistant professor of biomedical data science and by courtesy of computer science and electrical engineering at Stanford University. He works on making machine learning more reliable, human compatible, and statistically rigorous. And he's particularly interested in applications in human disease and health. And several of his group's algorithms are widely used in tech and biotech industries. He received a PhD from Harvard in 2014 and was at one time a member of the Microsoft Research, a Gates Scholar at Cambridge, and a Simons Fellow at UC Berkeley. And he joined Stanford in 2016 and is excited to be an inaugural Chan Zuckerberg Investigator and the faculty director of the university-wide AI for Health program. His group is also a part of the Stanford AI Lab, and his research is supported by the Sloan Fellowship, the NSF Career Award, and Google, Amazon, and Tencent AI Awards. So with that, we'd like to really welcome you, James, and thanks again for taking the time to join us. And if there's anything that you could maybe, by way of introduction, to tell us about in terms of how did you get interested in AI from the get-go? Because I think that's just one way to kind of break the ice here for our data science community.
Yes, sounds good. So first, thank you, Chirag and Murat, for hosting this, for inviting me. It's very nice to meet everyone from such diverse places. Yeah, so I've been working on AI and also this intersection of AI and healthcare biology for about the past 10 years. So my background was actually in math. So a lot of my undergraduate, a lot of my PhD work was in math, and I was very interested in more of like the mathematical foundations of machine learning algorithms and optimization algorithms. So I got into biology and healthcare when I was as a graduate student in Boston, where they let me hang around at the Broad Institute, where there's a lot of really exciting work on genomics, biotechnology that were being developed, right? So even though I was not trained as a biologist, they were kind enough to let me hang around and even do experiments. So basically, I spent a year, try to grow these iPS cells and then doing all sorts of perturbations on iPS cells and sort of injecting them into the brains of mice. So all the experiments failed horribly, but it was a great experience to learn about these really cutting edge technology tools like reprogramming, single cell analysis, genomics, and really think about how they relate to human disease or human health. So I came to Stanford in 2016, and here at Stanford, super fortunate to work with really amazing students and amazing collaborators. And here my group really has 2 main pillars, right? On the one hand, we're still very interested in working on sort of the foundations of machine learning, try to make these algorithms more reliable and just more efficient. And the other hand is that we're very excited about taking these algorithms really just out of sort of papers and labs, but really thinking about how do we deploy them in really high impact healthcare biomedical applications, right? So we're currently in the process of, for example, running several clinical trials here at Stanford in testing some of these AI algorithms. We've also been fortunate to work with large companies like Genentech, where we use different AI algorithms to try to improve the designs and efficiencies of clinical trials for cancer trials, and that's also being deployed and developed. So that sort of hopefully gives you a picture of the broader interest we have.
Well, yeah, thank you for that. I mean, it's really exciting to even hear that you were able to consider venturing into the biological sciences side with your background in mathematics. That takes some courage, obviously, as you saw firsthand about, you know, the trials and tribulations of doing that kind of research. So it's but really great, like you said, to give you that kind of hands-on perspective of what the some of the advantages and also potential limitations are, and how you could, you know, find space for AI to improve, you know, the use of the data that we have, and then also the design of clinical applications. So I'm really curious to learn more about, you know, how you're using AI to improve the design of clinical trials, because certainly that's one of the final hurdles we need to overcome before, you know, drugs or therapies can be approved for widespread use. So what are some of the areas there that you're able to work in?
Yeah. So I think clinical trial designs, it's a super exciting and really important space. It's also a space where there could be really high impact AI applications, but for a variety of reasons, usually related to data access, there hasn't been as much AI work in that area, right? So as you mentioned, the clinical trials, in some sense, is really the bottleneck for all of medicine, right? So all these great R&D research that we do here at Stanford, other universities, pharma companies, biotech companies, to really get them to patients, still the most common way is to really go them through the FDA clinical trial process, right? And each trial itself can be hugely expensive. Some of them will cost hundreds of millions of dollars to run, which is one of the reasons why typical overall development cost of drugs, you know, could be over $1 billion. That's why these drugs are so expensive to develop. So we've been working with our Genentech and Roche in thinking about this clinical trial space, right? And our starting point is to really think about how are these trials designed, right? So a big part of how these trials are designed is just in deciding which patients are eligible to even participate in a given trial, right? So let's say that I want to run like a breast cancer trial or a lung cancer trial, right? Usually they will have a huge PDF of, you know, over 100 pages that just describe what this trial is, right? And the important part of that PDF says that, you know, if you have this lab value, or hemoglobin counts less than this value, or if your bilirubin level is above this value, then you are or are not even eligible to participate in that trial. So I think that's actually a really big part of the trial design. And then we want to take more of a data-driven optimization approach in thinking about how do we think about deciding which patients are eligible to participate in the trial. And that, you know, in the beginning, that seems like a fairly esoteric problem for a computer scientist, but the more that we learn about it, it really became to us, like, I think, one of the really impactful immediate points, right? So one thing to notice that actually a large number of clinical trials in cancer space fail, right? Something like 40% of the trials fail to reach the minimum enrollment requirements. So they fail because they don't have enough patients. So each one that fails is hugely expensive, right? Millions of dollars. The flip side of this is that a large number of patients, right, over 90% of the patients with cancer will love to enroll in clinical trials because that's how they can get often access to life-saving cutting-edge technologies, treatments, right? Immunotherapy, targeted therapy, right? But then they're not eligible to participate because they don't fulfill these rules, right? These pages of requirements. So that's where we've been working on, you know, developing these algorithms to see, okay, so how can we take a data-driven approach to decide how to set these eligibility rules to balance two objectives, that we want as many patients who would like, you know, to be able to become eligible for these trials, right? But at the same time, we want to make sure that these patients are safe and the treatments are effective, right? To maintain the efficacy of these trials. That's something that people have been mostly trying to balance design more heuristically, but we thought, okay, this is where we actually have a lot of data and we can do this more systematically. And so in this work that we published with Roche Genentech, we developed this tool called Trial Pathfinder. So the paper was published in Nature earlier this year. It was led by Rixiang Liu, who's an amazing PhD student in my group here at Stanford. And what Trial Pathfinder does is that it actually goes through large medical records of hundreds of thousands of cancer patients and conduct a bunch of computational analysis to see, okay, so which patients have been given which treatment and what is their safety and what is their adverse events and efficacy outcomes. And based on all of that, then we are deciding then how to design trials for lung cancer, for breast cancer, for each of these cancer types. And the upshot of all of this is that we show that we could actually more than double the number of eligible patients, right, while still maintaining and even reducing the adverse events and reducing the hazard ratios of these trials. Right? So that becomes really a win-win for both the patients and for the pharma companies and drug developers. So just one example of where I think it could be a super exciting area of applying AI to make clinical trials more inclusive and more effective. Right? And inclusivity is also just one last comment, is also a big part of this, is that actually a lot of the patients that were excluded from clinical trials were minorities, older patients, more women. Right, so we show that by actually taking this data-driven approach that's less biased and make the trials more open to the broader public, we can recruit more of these minority, different demographic subgroups, more women, more elderly patients. That makes it actually potentially the broader benefits for the entire community of patients.
Well, it's really exciting. I mean, we know that the typical structure for these clinical trials is that you have a phase 1 for safety purposes of, first-time use of a new intervention in humans, and then you go on to phase 2 and phase 3 for efficacy. So would the idea here be to perhaps use some of these old, standard approaches where you have kind of almost generic, inclusion and exclusion criteria for the phase 1, just when it's, first introduced into humans, and then based on the results of the phase 1, then apply your AI approach to inform the phase 2 and phase 3 so that you can more rapidly and also more broadly include patients into that? Or is it to even perhaps somehow make the inclusion and exclusion criteria more flexible even from a phase 1 perspective?
Yeah, great question. So we are primarily focused on the later phases, so like phase 3. So we do use the information from phase 1. We also use a lot of data just from the real world data from the EHR, right? The idea there being that there are actually a large number of patients who are given these treatments, even if they would not have been eligible to participate in the original trial, right? Because the treatment after it comes on the market are given much more broadly, right? So then we can use those patients as basically a way to compute counterfactuals, right? To say, what would have happened if this person who's not eligible because their bilirubin level is off the chart, but what would happen if this person had been eligible to participate in this trial, right? And we can compute that counterfactual because we actually know what treatment is given to this person and what is their outcome.
Yeah, that's really exciting. I mean, just the ability to do this and use real world data, as you say, is critically important so that you can actually get a much more realistic sense as to whether you could, safely expand the criteria for inclusion into these trials. So I have a lot more questions I could ask, but we definitely want to involve the community as well. So one question that's coming in here from HW is, how can AI be used to minimize the use of animals in early drug trials?
Yeah, so that's also a great question. So I think there are two comments about this. One is actually related to the point of using the real world data, right? Like Chirag mentioned, I think this is really where there's this convergence, right, of a huge amount of data from EHR that's becoming better curated, and the R&D research stage for cancer drugs and for other drugs, right? So one of the things that I'm super excited about is actually using more of the real world data as a way to validate and to test a lot of hypotheses, right, before we really invest actual experimental resources on humans or on animals to do that. Right? So for example, a lot of drugs are actually used off-label in the real world. Right? So that means that the drug's approved for me for breast cancer and it's actually used on ovarian cancer patients, right? Even if it's off-label. And we have a lot of those data, right? We've been working with Flatiron Health and Foundation Medicine, which are basically collecting these large amount of cancer patients. And there we see that, oh, so here's like a cohort of cancer patients that were using drugs that are off-label, right? That are not officially approved by the FDA, but they're still showing strong efficacy because we followed them over 3 years, over 5 years, 10 years. So we know that these drugs, they're surviving and their lab values are looking good, right? So that actually becomes a great way to generate and to quickly test and validate a lot of hypotheses, right? That already demonstrate these drugs are safe on humans because people are already taking them, right? So that can help us to alleviate some of the usage of, of animal testing.
Yeah, I think that's wonderful. Being able to kind of streamline from the perspective of preclinical research and the burden on costs and time, and then of course also the lives of the animals that are being used to for the research, and then obviously to get to the direct patient population in a much quicker way is critical, because as you mentioned earlier in your introduction, a lot of the ways that clinical trials are designed is that they're based on large numbers. To get a statistically significant value. Whether that's a clinically significant effect is a different question, but as long as it's statistically significant, usually that leads to an approval of the agent. And as you're identifying, there may be a way to really find that ideal cohort of patients that doesn't seem to be as large, maybe an order of magnitude less than the standard, and still determine whether the intervention based on its molecular mechanism or, how it's thought to work, is efficacious for that particular subgroup. And then kind of feeding it forward, as you mentioned, once that becomes FDA approved for a particular narrow group of patients, it can then be kind of fed into the off-label usage and then continue to inform your work as well. So that's a really exciting way of kind of getting quicker approvals, quicker off-label usage, and then more feeding into your design here. So that's really amazing.
That's a great way to frame it. I just add one comment that I know that Murad would want to add as well, is that the other benefit of using the real world data and making these trials more inclusive is that the end product you get, right, the end drug that you get is actually much more reflective of how it would perform on real patients. Because one of the dirty secrets of clinical trials is actually people often who end up in these trials are sort of like the Olympic athletes of the patients, right? And then I was talking with some of my friends who was participating in one of these trials, and literally like the patients in that trial was like Kobe Bryant, a couple of Olympians, so like actually the Olympic athletes, right? And that's happens because the companies actually wanted to end up having like somehow like the healthiest among the patients that have these treatments, right? So then maybe shows the best effects. But you can imagine that something that worked on Kobe Bryant may not work on like a general population, right? So that's why I think there's just even for patient benefits, there's a lot of benefits in looking at more inclusive, broader data that comes from the real world.
Yeah, I mean, this is really interesting. We've been over 10 events, so this is the first time we're discussing about such topic. And it is a really important, big topic like clinical trials. Can you hear me well?
Yeah, yes.
Okay, good. So there are a lot of questions comes to my mind. For example, you do clinical trial. I mean, there are a lot of clinical trials and for different diseases. First of all, let's say some diseases, there are not much trials happen. For example, let's say very rare disease, MS, people do trials and there's not much data. But in terms of cancer, you guys have a lot of data in the past. So I'm saying there are a lot of bias in this clinical trials because it's disease specific and everyone reacts differently. First question, do you go disease specific, like a certain diseases? The second is your model can be applied in like in general or global, like a selection criteria?
Yeah, that's a great question. As a computer scientists, as a machine learning group, our natural tendency is to go as broad as we can to have as much data as we can. right? And so we're not specialized, we're not specialists in lung cancer or in breast cancer, right? So the initial analysis we did was actually across a variety of cancer types, right? So the lung cancer ends up having the most amount of patients, but we also validated in melanoma, breast cancer, colorectal cancer, and other cancer types. And I do think that some of the lessons about clinical trial design can be shared across different disease types, right? So for example, we did find that some of the laboratory values, right, that can be consistently relaxed across a variety of cancer types to make the trials more inclusive without compromising the safety or the efficacy of the treatment.
Also, there is another question, like you're getting data from companies or from the schools, like clinical trial data?
Yeah, so this is where we found it to be maybe some of our best collaborators actually the companies, right? So we get a lot of internal clinical trial data from Roche, which also owns Genentech. And Roche actually acquired these two other companies like Foundation Medicine and Flatiron Health. As Flatiron Health is basically like a data aggregator that goes across hundreds of cancer clinics in the country, in the US, so, right, from all the different states, and they aggregate all these medical records. That's how they have such a large data set.
So is there a chance that, let's say, Pfizer or Merck, they did similar study? Is there a chance to get their data, or just not possible to have a big data set?
Yeah, so in general, or in the past, I think sharing of clinical trial data across companies has been very, very sparse. They tend to be very protective of their data, which is also one reason why there has been relatively much less machine learning or data science work in thinking about how to optimize for the clinical trial space, right? Just because of the lack of publicly available data. But there are resources now that maybe would make it better to do this. And this is something that we're trying to push for as well. Like, for example, the tools that we developed, even though it's trained on the Roche data, but we actually made it all open source, right? So people from Merck or from smaller biotechs or people here at Stanford can already use these tools, because our goal is to not just to benefit one company, but really try to help the entire biopharma industry.
I think building on the comment you made about how the field that you're in really, of course, favors having more data than less to inform its work, and mentioning the kind of work you do with companies like Roche that now has acquired Foundation Medicine, which characterizes the tumor tissue itself. In your dream case scenario, what additional data would you want? Because you mentioned the time you spent at the Broad Institute learning about all the technologies that are going on. So what could further augment your work in terms of knowing about the patient's entire genome, knowing about their epigenome, and knowing about all the gut microbiome? There's so many things that are emerging to help us understand how, one drug may work in one patient with a particular disease, but it may fail in another because of something to do with the immune system or something else. So if you had your dream scenario and the costs were such that these things could be done routinely in patients beyond just testing their tumor sample, what other kinds of information would help to augment the ability of your work to get more customized approaches, let's say, for predicting what's going to work and what may not work for a patient?
Yeah, great question. I think the top of my dream list is sort of spatial genomics data. Right. So just to unpack that a bit more, so oftentimes we do collect some genomics data, right? If we're fancy, we can even try to collect sort of single cell RNA sequencing data from these patients. But most of the data is actually misses sort of the spatial information, because we have to sort of extract the cells, and then we don't really know where the cells are related to each other. So there's been several, I think, super, super exciting recent developments over the last couple of years in thinking about taking samples, biopsies from patients, and in the native state of these samples, then sort of measuring spatially resolved genomic information or protein information at the single cell resolution. Right. So we did some work in this area. There's a really excellent computer science PhD student in my group, Brian, who developed one of the first methods to look at spatial transcriptomics using computer vision algorithms. Right. So I think that's an area that's really starting to explode. Right. I think spatial genomics was, I think, named by Nature Methods to be like method of the year in 2020. Right? So this is very much happening now. And some of our current efforts are also leveraging spatially resolved protein information, right? That's also at single cell resolution. So that's then becomes super exciting, right? Now you can imagine you take a biopsy sample of a patient, you get maybe 1 million cells, and now we know exactly this type of each cell, right? Because we have single cell protein information for 40 or 50. So these are multiplex stains, right? So we get 40 or 50 markers. So it's much richer information than people had done previously with pathology slides. And we also know very fine-grained subtype of these cells because we have so many multiplex markers. And since we know exactly where the cells are related to each other, we know for each tumor cell, like how close is it to different immune cells, right? And we also know how exhausted the immune cells are, how active they are. So that gives us like a whole new dimension of information that's really, really relevant to cancer patients, right? And especially for things like the new treatments like immunotherapy, I think having this spatial view where you know where exactly the immune cells are, what exactly each cell is doing, and how close they are to the tumor cells can potentially be really transformative for both biomarker discoveries, for target discoveries, and also just for predicting patient outcome.
So that will be, that is fantastic, but it will be very costly. Do you guys have study like that? You do also single cell genomics of the patients' samples?
Yes, I would say these are still in the research stage. So a couple of the projects in my group, also in collaboration with companies like Enable Medicine, have been looking at this, where we have thousands of patient samples from several different diseases, right, from different cancer types, also non-cancer studies, where usually we'll have on orders of maybe like 100 patients, right, per disease. And then we get a few samples from each patient. And the really nice thing is that each sample you get from one patient is so rich in information, right? Because you can have like 100,000 cells in their native states, in their native location. So even each patient sample gives you a whole terabyte of information that we can then train our machine learning algorithms on.
There are ocean of data. Like, I can't imagine one patient will give you a lot of data. Having genomics, RNA data, and single cell genomics will be actually something different. Proteomics is a huge data. And it would be interesting to see results from that. There's one question before I let the others questions. So do they provide you failed trials so you can use it in your model? Because I know there are a lot of failed trials that people don't even know about it. So.
Yeah, so we have looked across like a diverse set of trials. So we have trials that are successful, they failed. We also have sort of more fine-grained information in between, right? Because even successful trials, sometimes they just they end up taking too long to enroll patients, or they end up having very high patient dropout ratios for some reasons. Right, so actually one of the ongoing projects in the group is to look at these more fine-grained operational metrics of each trial, right? Not just success or fail, but just how long did it take to enroll patients? What is the patient dropout rate, right? What was your initial targeted number of patients that you want to enroll, and how long did what is the number of patients you actually end up enrolling? Right, so these more fine-grained operational metrics are something that we have access to, and we're building models, machine learning models, to be able to say, okay, can we actually forecast and predict those operational metrics?
One question came up again. If you're doing phase 3 studies of phase 3 studies, clinical trials. So like based on phase 1 and phase 2 response of that therapy, do you have a predictions of phase 3 will be successful or not, or you didn't go in that direction?
Yeah, so that would actually be the holy grail, right? Because if you could do that, then you could potentially save billions and billions of dollars, right? If you have like an oracle that tell you which one's going to work. So we're not quite there yet. So we decided first to tackle these more operational metrics as a way of getting there, right? So something that's more quantitative, like, okay, how likely is this trial to enroll patients, right? So that doesn't quite say how effective the drug will be on those patients, but at least we can get you like how these efficiency metrics. I think it's super exciting area going forward as more data like this become available to then to really to solve the holy grail question of, can you then say, how can I prioritize, have an algorithm to prioritize, given the phase 1 the information and the real world data, right? Which drugs are most likely to be effective in their phase 3 studies. So not quite there yet, but hope to get there in a medium horizon.
That's really exciting. Particularly in the field of brain cancer, particularly glioblastoma, there's a lot of excitement, as you mentioned, for immunotherapy and other solid organ cancers. But all of the phase 2 trials of immunotherapies as single agents in patients with glioblastoma have looked promising. And then they all fail, unfortunately, once they get to the phase 3. And so the work you're doing is critically important to, down the road, to be able to kind of help inform us early on, if we can, whether it is a success or a failure, just so we have less patients than even put onto trials that may, be futile, for example. So this is really exciting work. And this segues into 2 related questions from the community.
I'm sorry, it's related to yours.
Okay.
Either one thing came to my mind again. So in terms of, for the glioblastoma, for example, so there's an IDH1 mutation, and that changes the effect of the therapy. Is that right, Shaq?
So the MGMT promoter methylation.
Okay.
Yeah, so the IDH, so there's the IDH mutation status of the patient's tumor is more prognostic in general, whether there's going to be a longer short term. But then if you look at the epigenetic information of the methylation status of this MGMT promoter, that will predict more or less likely responsive to the particular chemotherapy that we use, right?
So let's say you guys are looking at genomics and all this data, and then you figure out that some type of a mutation or like a isoform of the protein's existence makes the therapies different. But what if that causes a problem? You go back and choose the patient the way you want it, and then it increases your clinical trial accuracy rate or efficacy. Do you think that could be a problem?
Before James answers, I think that's a great thing, because actually we are looking for the patient cohort that is enriched to succeed. And so, the current standard of clinical trials is to say that we're going to take patients with glioblastoma and give them a therapy. If we could say that we're going to take patients with glioblastoma who have this gene mutation and give them this specific drug, then even if it's a small cohort of patients, as long as the drug has high efficacy, that's still a win, for that particular subgroup. And I think this really gets to this whole idea of a one-size-fits-all approach not being applicable to tumors. And even to James's point about these types of massive data sets he's getting from Foundation Medicine, now in cancer, it's not as important, perhaps whether the tumor is coming from the breast, the lung, the brain, or wherever. If it has a particular mutation status of a particular gene, it may be rendered susceptible to a drug, regardless of where it's located in the body based on some molecular characterization. So I would argue that indeed, if we can find out information about, why is this patient responding or not responding based on a particular gene mutation that we're not aware of right now, and that can be validated prospectively, that new knowledge can really help us to guide our therapy. So, but I'd definitely be interested in James's take on that.
Yeah, no, I agree. I think that's a very good comment. I mean, I think, especially for diseases that are so heterogeneous, right? You can have drugs that sort of fill on the, applied on the aggregates, but it can still be successful on the subpopulations, right? And I think it's really exciting to think about how do we find markers that can be used to identify subpopulations for which a given treatment is likely to work.
Yes, absolutely. There's a couple of related questions here. One is, when do you see the digital clinical trials being actively implemented in clinical trials strategies, broadly speaking? And then the second one is a little bit more specific to the earlier part of your talk, which was, the question is, your algorithms create an enhanced patient population for clinical trials, and then the trials have to be emulated with this new enlarged population or enriched population. How reliable is this emulation?
Yeah. So both are great questions, and they're definitely related to each other. So Yes, so our strategy is to emulate trials with basically to create a large number of emulations of trials by using these real world populations. And I guess the way that I like to look at it is that, traditionally you only get one shot on goal, right, in these clinical trials, right? Because each trial is so expensive, you just run it once, maybe you already spend $100 million, and then it would be in my ideal world, you actually run a bunch of these trials in parallel, but you tweak, changing different conditions, right? That's okay for the patient with this biomarker, but you tried. You have one cohort, patient with biomarker B, you have a different cohort, patients with bilirubin above this threshold, right? You can try all these different combinations. But in reality, you only get one shot on goals. You have to pick one of these to run, right? So the whole idea of these digital clinical trials or the trial emulation is now, instead of getting one shot on goal, maybe I can take 1 million shots on goal, right? Because I can computationally, right, create 1 million of these different cohorts. Each of them defines a subset of patients I see in the real world, right? So they're synthetic cohorts, but it's really coming from the real world patients, right? And then I can see, okay, I have on my computer, then I can see, okay, how would all of these millions of cohorts perform, right? And then we can try to come up with the algorithm to summarize the information, the simulations from 1 million trials to help us make recommendations. I think the question is correct that at the end of the day, we still need to run this trial on the real world, actually like a clinical trial on new patients prospectively, right? But just now that if we have these millions of synthetic trials that we run, right, that gives us a lot more confidence about how to design the actual really expensive new trial, right? So that's where I see this interplay is and why there's very complementary.
Yeah, I think that's critical inference, almost kind of like in certain, fields of science or statistics, they talk about Monte Carlo simulations to try to, kind of play out what may happen. But here you're kind of doing that pre the actual clinical trial, in which the big investment of funds and patients, needs to be placed to actually then answer the question. That's great. So yeah. I guess a follow-up to that is, do you ever envision that, the FDA may get to a point in, years or decades from now where they may even approve, drugs based on a virtual trial basis, or is it always going to have to come down to implementation into actual patients and getting that kind of survival benefit or meeting of a primary endpoint?
Yeah, that's an interesting question. I think the FDA is becoming more and more receptive to data from these digital analysis from real world cohorts from emulated trials, right? And we've also had some good discussions from the FDA folks. And I think there's sort of two converging trends from the FDA side, right? So first, the FDA, they themselves recognize that existing trials are just way too restrictive and they need to be applied to broader populations, right? So that's actually one of the mandate of the FDA, that's all these cancer trials needs to be designed in a way that's more inclusive and more data-driven, right? So already they're very much in favor of the kinds of AI algorithms like exactly the ones that we're proposing here. And the second trend is that, they are actually, I think, very also becoming much more receptive to the real world data, right? I think before she stepped down, I think, Amy Abernathy, who's one of the second in command at the FDA, she was actually also the leader at Flatiron Health, right, before she joined the FDA. And now she recently stepped down over the past year. Right, so you can already see that, the people who are really leading the charges at the FDA have also been coming up through this pipeline, thinking about very thoughtfully about the real world data and the benefit of real world data, and who have even created, I think, some of the largest such data sets that we use to emulate trials. Right, so I think there's going to be like a confluence of factors that will make the FDA more receptive to this kinds of data. I think still probably what will likely happen will be some sort of hybrid model, right, where they still want to see maybe some smaller scale new prospective clinical trial in combination with data science, AI-driven recommendations from large patient cohorts, maybe from the real-world data.
That's amazing. And I think another question that commonly comes up at our talks is, the large majority of the audience here are data scientists. And so you have this really unique background starting in mathematics and then kind of your curiosity peeking into biology and kind of putting yourself in the Broad Institute and learning about that, and then moving on with your career. And now you have a lab full of folks who are doing data science, but really with this unique data set, and you kind of made those connections to make that happen. So what advice do you have for the data scientist community here that are really interested in getting involved with biomedical research? What advice do you have for them in terms of how to interface with biological researchers or clinicians to take their skill sets and help apply it to the benefit of science in general or clinical medicine?
Yeah, it's a good question. I mean, the first thing I would say is that this is a super exciting area, right? This intersection of data science and AI on the one hand, and healthcare, biomedicine, right? Especially with the pandemic and all of this. I mean, this is like the most exciting area that everyone's interested in, right? So even the traditional tech companies like Google and Amazon and Apple are making huge investments exactly at this intersection, right? Because everyone recognizes that this is where the transformation, the next big breakthroughs will happen, right? And the second point is that I think we're also in this area where a lot of the data-driven algorithms, I mean, there's enough data sets available where a lot of the innovations on the algorithm side and then the methodology side can be really transformative to the biology and to the medicine, right? Like, for example, AlphaFold from DeepMind is a great example of this, right? I mean, they're leveraging mostly publicly available data sets, right? There's large protein data banks, but they also leverage these quite impressive compute resources that they have at DeepMind, which is also owned by Google, right, to develop these AlphaFold algorithms to solve really, but at the end of the day, they solved, made a huge amount of progress in something that's really transformative to medicine, right, which is looking at the protein structures, right? So that's a confluence of a lot of publicly available data that people have curated, plus really interesting algorithms, right, making progress. I mean, our work on the clinical trial design has similar flavor, right? It's a lot of data that's coming from the medical records, right? With a lot of compute resources, now we can start to do something like optimizing clinical trials that people just didn't have the resource to do before, right? So I think thinking about, I mean, my advice would be look for areas where there's a lot of new data being produced, right? And then also areas where there's a lot of high capacity models, right? And I think that's usually a good intersection where there's a lot of new things to be made, new innovations.
So yeah, thank you for that. That's really very helpful. Another question is in regard to some of the privacy issues or the HIPAA laws, et cetera, that you may run into in order to be able to access a lot of the patient's data from these electronic medical records to help inform some of the real world, to curate at least the real world information of what's going on with off-label usage of drugs. What kind of challenges do you face in that regard? Because certainly in order to make a great model, you need this kind of data, to the best of ability in an anonymized way. So you obviously protect the patient's privacy and, reduce the risk for any kind of unnecessary sharing of personal health information. What have some of the challenges been that you've come across in that regard?
Yeah, so I think that's a great question. And actually a core part of my research here at Stanford is really thinking about this data-centric perspective onto medical innovations and also AI development. Because for us, oftentimes the biggest challenges going forward is really thinking about how do we get the right data sets into the right algorithms. Right? So algorithms, once you have the data, algorithms oftentimes are even easier to develop, but really thinking about how to get the right data together in compliance with these privacy laws and other regulations, that's often the biggest challenge in practice. So there are lots of really interesting techniques here, right? So something that we've been exploring would be having settings where the data stays in one place and then the model can be moved very quickly, right? So one of my students, Abu Bakr, developed this tool called Radio, which basically makes it very easy for the model to be shared. So the data can stay in one place, right? It stays at your hospital or your clinic, and the model can be shared as easily as someone just copying and pasting like a URL and opening it on their browser, right? So then it's actually Radio, it literally creates like a URL, you can open up on your browser, and then you immediately would have access to the very sophisticated models that your collaborator or colleagues have developed. Right, so I think these kinds of tools that enables data to stay in place, or data to be centered around in secure private formats, but the model to be very easily moved around, right, to these other places. I think could be one paradigm going forward, especially since that there are very strong, as you mentioned, very strong regulations about data being moved, like in GDPR in Europe. That's one such regulation. Certainly data from countries like China, it's been very hard to move outside of China, right? So there's a lot of interesting reasons why now data has to sort of relatively stay within some physical borders, whereas there hasn't just been such constraints about models, right? I can still send my model to colleagues, in Europe, to China, so there's no constraints. I mean, it's just sending a URL or sending links, right, to some models. So I think thinking about ways where we can really leverage the fact that the model can travel freely without border as a way to mitigate the fact that data has to stay within borders.
Well, that's really exciting. That really helps you then to open up. And I think that you raise another important point, because certainly when clinical trials are done in certain countries or certain geographic areas with certain ethnic populations, they are sometimes validated or repeated in other countries or in other ethnic populations to confirm the validity of those results, so that you can kind of have general applicability. And so by being able to then transfer your model, as you say, across borders, so that they're kind of being applied to individualized data sets, will just push this forward even faster. So that's really exciting. One question from the community here is a more general question that says, classical counterfactual reasoning is still at the heart of causal inference in the trials. Now that we have access to big data, can causal inference be done any differently?
Yeah, so that's a very interesting question. We actually use quite a lot of causal inference in our machine learning works and platforms, right? I agree with the question that's at the heart of it, right? To do a clinical trial, you really want to test like a causal relationship, like, does give treatment A causally lead to better outcomes compared to giving some existing state of treatment B, right? Previously, it's been very hard to actually do causal inference without having to do actual experiments, right? Ideally, to test a causal question, you have to do this kind of A/B test, randomized clinical trial or A/B test experiments. We actually benefit a lot from using these very large real-world data that enable us to basically simulate A/B test experiments, right? And the way that we simulate them is that now that we actually collect so much information about each patient and such rich amount of information from each patient, that enables us to actually try to correct for all sorts of potential confounders much more effectively than before, right? So the intuition there is that if we don't see anything about the patients, then I don't know why they're given treatment A or treatment B in the real world, right? Maybe there's some very strong preference, like sick patients are given treatment A and healthy patients given treatment B. If I don't know their sick or healthy status, then I can't really correct for that confounder. That's why it'd be hard to emulate a real trial or emulate a counterfactual analysis using observational data, right? But if I actually observe their lab values, all of their ICD codes, right, their heart rates and all of these other information, then I can use that high dimensional data to effectively try to correct for these potential biases, right? That actually makes it much more effective and more easier to emulate counterfactual reasoning using observational data just purely through these algorithmic approaches.
That's great. We are actually getting close to the end of the panel. So I have last few questions, and then this will be the last. So one of the things that one thing I should tell you, the Gradio app, I shared the link, and your lab got my attention after listening that talk in Ames. It's a fantastic idea. I listen, it's a great app, great idea. I hope it goes much, better directions, and then it will be useful to everyone. I like the concept a lot. So that's why I shared it. And the one thing is, people ask usually, do you use like open source data? Did you try those data, and do you know you have references for those open source places that people can use? Maybe we have a lot of data scientists in our community.
Yeah, so we do use a lot of open source data. So especially in biology, there's actually a tradition of very good open source data, like the TCGA for cancer, for example, is actually all open source. There are also other places like, for example, AWS Data Exchange, which are basically third-party data platforms where there are thousands of data sets on there, some of which requires money, but others are actually free, right? So you can just download them. They're provided, for example, for like COVID-19, there's actually a lot of available data on AWS Data Exchange, right? So I think those are useful resources for data scientists because they have a lot of very large, interesting data sets.
That's great. So actually, do we have the questions very similar to earlier ones? So thank you for being here today. It is nice. We connected the world with the Zoom, through the Zoom and through this event. Appreciate everyone's time and your time, James. It is an honor for us to host you. I appreciate it.
Yeah, thanks for the great discussion.
Yeah, thanks for the great discussion and questions.
Okay, good to meet you. Okay, take care. Thank you. Okay,
thanks everyone.
Okay,
bye.
Bye.
Take care.